Hello everyone, welcome back to Allied Bites. Today we are going to study about morphology and physiology of bacteria. The same book I am referring, Essential of Medical Microbiology by Apurva S. Shastri. What are the learning objectives? At the end of this session, the students will know the classification of microorganism, to understand principles of microscope and staining techniques, differentiate between gram-positive and gram-negative bacterial cell wall appendages, and other structures of physiology of bacteria. Then, the first coming for characteristics of prokaryotes and eukaryotes. As you all know, what do you mean by prokaryotes and eukaryotes? Just I will give a brief introduction about prokaryotes and eukaryotes. What is the major group of prokaryotes? Bacteria, blue-green algae. Eukaryotes, fungi, parasite, other algae, plants and animals. Coming for the nucleus, prokaryotes has diffuse nucleus, eukaryotes has well-defined nucleus. Coming for nuclear membrane, in prokaryotes, nuclear membrane is absent. In eukaryotes, nuclear membrane is present. Nucleolus and ribonucleoprotein are absent in prokaryotes, which are present in eukaryotes. And cell division occurs in prokaryotes through binary fission. In eukaryotes, mitosis and meiosis. Then coming for chromosomes, prokaryotes has one or circular chromosomes. Eukaryotes has many, but it is linear. Then coming for extra chromosomal DNA, in prokaryotes only found in plasmids, eukaryote found in mitochondria. This is about characteristics of prokaryotes and eukaryotes, which you already know. Moving for the next slide. Coming for the cell membrane, doesn't contain sterols except in mycoplasma. That is about prokaryotes. Eukaryote contains sterols. Okay. Cellular organelles such as mitochondria, Golgi complex, ribosome, everything is absent in prokaryotes and present in eukaryotes. Ribosome 70S size in prokaryotes and 80S size in eukaryotes. Size of respiration, mesosome is the site of respiration in prokaryotes and eukaryotes, mitochondria. Pinocytosis is absent in prokaryotes, present in eukaryotes. This is about prokaryotes and eukaryotes difference. Then coming for microscope. What do you mean by microscopy? What do you mean by microscope? We shall discuss in detail about microscopy and microscopes. Coming with the first thing, that are properties of microscope. Properties in the sense to have, it should be, if any of the microscope, if it is should be a good means, it should have some properties. Mainly we have three properties. In that, the first thing is good resolution. What do you mean by resolution? Resolution power is an ability to produce separate images of closely placed object so that they can distinguish as two separate entities. Okay, the oil immersion has higher refractive index than air, which enhances the resolution power. For example, if in the human eye, resolution power will be 200 micrometer. In the light microscope, 0.2 micrometer. In the electron microscope, 0.5 nanometer. Okay, this is about resolution. Then contrast. What do you mean by contrast? Color. Okay, it can be improved by staining the specimen. When the stain is binds to the cell, the contrast is increased. When we give color, so you can improve the contrast. Okay. Then the last one is magnification. This magnification is achieved by using the lenses. Ocular lens with a magnification power of 10x. Then objective lens with the objective lens used for scanning, that is 4x, and low power 10x, high power 40x, and there will be oil immersion, that will be 100x. Total magnification, how we are going to calculate, means magnification of objective lens into ocular lens. Magnification of scanning field or low power, high power and oil immersion field is 40x, 100x, 400x and 1000x respectively. These are the properties of microscope. Then coming for the microscopes, here we have classification in the microscopes. we have different types of microscope which we are divided into light microscope and electron microscope. In light microscope we have simple microscope, compound microscope. In compound microscope we have dark field microscope and bright field microscope, and then phase contrast microscope, fluorescence microscope. Where coming for electron microscope, it has transmission electron microscope, scanning electron microscope. This is the classification of microscopes. Okay, then what are the mechanical parts of the microscope? Means it has a base. Can you see the base here? This is a base, and this is a C arm, C shaped arm, and this is a mechanical stage. These are the main important parts of mechanical parts of microscope. What do you mean by simple microscope? It has double convex lens with a short focal length. This lens is a single high powered converging lens that produces vertical images by magnification, which cannot be projected on screen like a real image. Okay, for example, Dutch scientist Antony van Leeuwenhoek, he used magnifying glasses. They are like a simple microscope. Then coming for compound microscope, in this we are going to use two or more lenses to magnify the specimen. Not by only the single lens, we are going to use two or more lenses. In that, the first thing is bright field microscope. What are the magnifying parts of this bright field microscope? Means ocular lens, objective lens. Can you see here? Ocular lens. It is mentioned everything here. Then objective lens, condenser is there which focuses the cone of light onto a slide. In that condenser you can see iris diaphragm, light source, and fine and coarse adjustment knob. Everything is mentioned in this picture. You can see it clearly. Then what is the working principle of this microscope? Means it is the most commonly used microscope. Image of the specimen is formed bright and colorful, and the background is also bright, but this requires staining. Okay, but the principle of this microscope means based on the use of visible light and objective lenses to magnify and illuminate the sample. Okay, when a sample is placed on a stage of a bright field microscope and illuminate from the below, the light passes through the sample and focus onto our objective lenses via condenser lenses. Okay, this is the working principle of bright field microscope. What is the application of this bright field microscope? Means biology and medicine. Used in the biology and medicine, used in pharmaceutical industries to identify contamination and impurities in the drugs. Then coming for dark field microscope. Before going to dark field microscope, we shall learn about what are the advantages and disadvantages of this bright field microscope. It is used in research and educational labs. Brightness of an image can be easily adjusted using the light source, and optics which do not alter the color. Coming for the disadvantages of bright field microscope, it can't view bacterial cell. It has low contrast, hence most specimen must be stained for them to be visualized. Staining may introduce unwanted detail into specimen. No need stronger light. Okay, for this light field microscope we need stronger light. Then the next thing is dark field microscope. This dark field microscope, objects are brightly illuminated against the dark background of a microscopic field. Whereas in bright field microscope, the background will also be bright, but here it will be dark. It uses special condenser and objective lens to scatter the light around the sample, which creates the bright image against the dark background. Coming for the principle of this, it uses light microscope with extra opaque dark underneath the condenser lens, or a special condenser having a central blacked out area, due to which the light coming from the source cannot directly enter into the objective. The path of light is directed in such a way that it can pass through the outer edge of a condenser at wide angle and strike the sample to an oblique angle. Okay. Only the light scattered by a sample will reach the objective lens for visualization. All the other light that passes through the specimen will miss the objective. Thus, the specimen is brightly illuminated on the background. This is about the working principle of dark field microscope, which is given in the picture. You can see that picture. Okay, coming for the applications, it can observe living and unstained cells. Internal structure can be seen and used for the study of external structure in detail and used for the study of motility. Coming to the advantages, no need to stain, resolution power better than the bright field, image contrast without use of stain, and thus do not kill the cells. What are the limitations? They are very sensitive to dust. Sample must be very strongly illuminated, which can cause damage to the sample. This is about dark field microscope. Then coming for phase contrast microscope, it is similar to like a dark field microscope, but cone of light passes through a cell. Some light rays are bent due to the variation in the density and the refractive index within the specimen and are retarded by about 1 to 4th of wavelength, 1 by 4th of wavelength. Okay, this undeviated light rays strike a phase ring in a phase plate, while the deviated rays miss the ring and pass through the rest of the plate. It is same like in compound microscope, only the addition is annular ring and the phase plate. Okay, what is the principle of this? The phase contrast microscope separate the illuminating background, the specimen scattered light. The phase contrast microscope is used to visualize the transparent, colorless, unstained living biological specimen. This is called as phase object. Okay, this is called as phase object. What are the applications of this? To use it for studying microbial motility and determine the shape of living cells and detecting bacterial components such as endospores and inclusion bodies. This is about phase contrast microscope. Applications are given in detail in these slides. You can take down or you can take a screenshot of this. Then next is fluorescence microscope. It Fluorescence uses fluorescence property to generate an image. It is a source of light is mercuric lamp. Emitted rays pass through an excitation filter will only allow short wavelength UV light of about 400 nanometer to pass through. Okay. Then excited rays get reflected by a dichromatic mirror and which will falls on a specimen which is stained by fluorescent dye. Fluorescent dye is nothing but they emit like a lights. Okay, the color which emit like a light. Can you see that in a picture like green or yellowish thing? Can you see that? Then barrier filter, it removes the remaining UV light which damages the viewer's eye, or blue or violet light which reduces the contrast. Okay, then coming for applications to demonstrate the autofluorescence property and get the better resolution. Then coming for electron microscopy. In electron microscopy, as I already said, we have two types. One is transmission electron microscopy and another one is scanning electron microscopy. They both uses accelerated electrons as a source of illumination. Okay. Here that given the features of the difference between the light microscope and electron microscope, you can go through that. No need of explanation and all, I guess. Moving for transmission electron microscopy, specimen preparation. Very thin specimen, that is about 20 to 100 nanometer thickness is needed. Bacterial cells, they are sliced into thin layers. To prepare the thin specimen, the following steps are needed. What are the following steps? The first thing is fixation. Cells are fixed by using glyceraldehyde or osmium tetroxide for stabilization. Okay. The next thing is dehydration. Specimen is then dehydrated with acetone or an ethanol. Then the third thing is embedding. Specimen is embedded in a plastic polymer. Then the slicing. Specimen is then cut into thin slices by an ultramicrotome knife. Such thin slices of specimen are mounted on a metal slide or a copper. Okay. This is about with the steps to get a thin specimen. Then coming for the principle, it is similar like an light microscope. Light microscopy uses light rays to focus and produce an image, while the transmission electron microscope will uses a beam of electrons to focus on the specimen to produce images. Electrons have shorter wavelength in comparison to the light, which has longer wavelength. Okay, coming for the applications, it helps for visualize and study the cell structure of bacteria, viruses and fungus, and view bacteria, flagella and plasmids, and view shape and size of microbial cell organelles. What are the advantages? Powerful magnification of 2 million times that of light microscope, used for ranging from basic biology and nanotechnology. It produces very efficient, high quality images with high clarity. What are the limitations? It is very expensive and it is big to handle, and specimen preparation is very tedious and it requires constant inflow of voltage. Then next is scanning electron microscope. It scans surface of microorganism that uses a beam of electron moving at a low energy to focus the scan specimen. What are the principles? Applying kinetic energy to produce signals in the interaction of electrons. The secondary and backscattered electrons used to produce an image. The secondary electrons are emitted from the specimen, play the primary role for detecting the morphology and topography of a specimen, while the backscattered electron shows contrast in the composition of elements of the specimen. Coming for the application, it is used in industries, nanosciences, biomedical studies, and used in analysis of cosmetic components which are tiny in size, and used to study filament structure of microorganism. And advantages: easy to operate and user friendly, used in variety of industrial applications. What are the limitation? It is very expensive and it is very bulky to carry.